we need to at least understand the few types of bases that you can find in the nucleotide. You can see that there are 5 types of different bases. So these are the nitrogenous bases. The nitrogenous bases. So these bases are the rectangle that we use to represent the nucleotides. So there are 5 of them that you need to learn. The adenine, which we use an A to represent it. So A. And then G, which is the guanine. And then U, which is the uracil, the T, which is the thymine, and then the C, which is the cytosine. You don't have to memorize or even know, like, how do we draw an adenine, guanine, uracil, thymine, or cytosine, but you need to at least find a pattern in here. You can see that we can actually divide these 5 bases into 2 groups, right? If we cut it up half, the top part, you'll realize that there are 2 ring structure in the adenine and guanine, right? So I'm going to draw out the ring here. You can see that there are 2 rings in adenine or guanine. It looks like rings that have been fused together. So when you have 2 ring structure in the bases, we call it a purine. So purine, P-U-R-I-N-E, purine has 2 ring nitrogenous base. And the one with only 1 ring, so you can see that this is 1 ring, this is 1 ring, and this is only 1 ring as well, right? So the 1 ring nitrogenous base is what we call the pyrimidine. P-Y-R-I-M-I-D-I-N-E. So pyrimidine only has 1 ring. So pyrimidine is actually 1 ring nitrogenous base. You're going to realize that 1 ring is going to be shorter, right? Short. Then the 2 ring structure. The 2 ring is going to be twice the length of pyrimidine, right? Because they are much longer. They have more rings than a 1 ring structure. So this can actually create can affect the shape of the DNA. If we look at it, DNA, we always draw it in a double helix shape, right? Where the bases connects at the middle part area. So right now I'm drawing is the DNA, typical DNA format, and right in the middle is actually the bases that we learned just now, the A, T, G, and C, right? Adenine, guanine, cytosine, thymine, and uracil. They are the bases that you find in the nucleotide. To make it clear, uracil can only be found in RNA, while thymine can only be found in DNA. So in DNA you can only find thymine, while in RNA only uracil can be found. So you always have 4 types of bases in either DNA or RNA. DNA has adenine, guanine, cytosine, and thymine, while RNA has adenine, guanine, cytosine, and uracil. So DNA has adenine, guanine, thymine, or cytosine, right? And they have to pair together. They can be paired together in order to form this double helix structure. This backbone here, we call this the phosphate sugar backbone, which is this group, right? You can see that this is the backbone of the molecule, and they have your phosphate group. So this is your phosphate, and this is your sugar. What makes DNA different from other things is the bases in between, right? So you can see that now we have this strain, right, which is a strain of DNA, is going to pair up with another strain of DNA, right? So they pair up by forming bonds between the bases. So you can see that this is a 2-ring base, and this is a 1-ring base, and this is the 1-ring base is going to pair with another 2-ring base. This is a 2-ring, always pair with 1 ring. So you can see that 2-ring always pairs up with 1-ring base. 1 ring always pair up with 2 rings base, so that the total will always be 3 rings. This is to maintain the distance of the 2 backbones, so that it will always be consistently far away from each other. If you have 2 ring plus another 2 ring, you're gonna have 4 rings in between. That is going to make the backbone fatter, and it will not have a consistent shape, right? Or 1 ring plus 1 ring is going to add up only to have 2 rings. That's going to make the whole structure a lot skinnier. So your DNA is not going to be smooth. So we want the DNA shape to be consistently wide. So you always have 3 bases in between the 2 backbone of the DNA molecule. We learned that A and G are 2 ring, while T and C are 1 ring. The purine will always have to pair up with pyrimidine. So the pairing method is A will always pair up with T, while G will always pair up with C. So this is the thing that you need to memorize. A has to be paired up with T, and G has to be paired up with C. We call this the complementary base pairing system. It is to maintain the shape of the DNA so that it will always only have a 3-ring distance. So complementary base pairing. A always has to pair up with T, while G always has to pair up with C. So you can see that here. Another one last difference between DNA and RNA, we mentioned the differences between DNA and RNA just now, is that RNA has a U ratio while DNA has a timing. The 3 bases, the 3 remaining bases are the same. They both have cytosine, they both have guanine, and they both have adenine. DNA is a very stable structure because it needs to store the genetic information in your body. So it's always in this double helix shape, right? So it's always in the double helix shape, and the pair is always in the middle, right? So you can see that this is the double helix pair. You can see the red is the T, right? This red base is the T. The timing is always going to be paired up with the yellow, which is adenine, right? If you keep looking down, the A will always have to pair up with the T. The red always had to pair up with the orange, while the green always has to pair up with the blue, because the green is the guanine and the blue is the cytosine. RNA is always single-stranded. It's only just one strand. So RNA doesn't form double helix structure. Of course, there are exceptions in different organisms, but at least in the human body, RNA does not form double helix structure. Only DNA does. And RNA is single-stranded. It means it's only one strand. You see that there's no counterpart that makes it a pair, right? RNA is not very stable, so they can get destroyed and just get decomposed in the cell. And that's all about DNA and RNA. See you in class. Bye-bye.